We're going to learn about the mechanisms of neural tube formation. Neural tube formation is one of the main processes that occurs in neurulation. As shown in the figure, the neural tube is a hollow tube-like structure that is formed, and there are three different ways by which it can form. The first mechanism of neurulation is called as primary neurulation, and this occurs in the anterior part or the rostral part of the neural tube. The second mechanism is called secondary neurulation, and this occurs in the posterior end of the neural tube or the caudal part of the neural tube. The third mechanism is called as junctional neurulation, and junctional neurulation occurs at the boundaries of primary and secondary neurulation, so it is usually observed somewhere in the middle of the neural tube. Neural tube formation is one of the main processes that occurs in neurulation. As shown in the figure, the neural tube is a hollow tube-like structure that is formed, and there are three different ways by which it can form. The first mechanism of neurulation is called as primary neurulation, and this occurs in the anterior part or the rostral part of the neural tube. The second mechanism is called secondary neurulation, and this occurs in the posterior end of the neural tube or the caudal part of the neural tube. The third mechanism is called as junctional neurulation, and junctional neurulation occurs at the boundaries of primary and secondary neurulation, so it is usually observed somewhere in the middle of the neural tube. So we're first going to look at primary neurulation, and primary neurulation occurs in 4 steps. The first step is the elongation and folding of the neural plate. So the neural plate cells that are right above the notochord, they start folding. And the presumptive epidermis, in this case, is also required for the process. So when we look at the picture that's there in the blue box, then here we can see that the neural plate is undergoing elongation and it starts folding. So we can see certain parts of it come up, and in this case we also have the presumptive epidermis also play a part in primary neurulation. The second step is bending of the neural plate. So in this case, a hinge point is formed called as the medial hinge point. Now when we look at the figure in the blue box, which is right at the bottom, this is where we're seeing the bending of the neural plate. And right in the small blue box is where the medial hinge point is formed. Now where this medial hinge point is formed, we can see that cells of the ectoderm actually are interacting with the notochord. And this can be seen in the smaller blue box that we see to the right of the figure. So the yellow cells are the medial hinge point that is contacting the notochord. Now we're going to see that there have to be changes in cell structure in order for this interaction to occur, but the formation of the medial hinge point allows the bending of the neural plate. It forms what's called as a neural groove, and this is formed at the dorsal midline. The next step involves the convergence of the neural folds. So the way we saw the medial hinge point, in this step we have another type of hinge point formed which is called as the dorsolateral hinge points. And these dorsolateral hinge points allow the neural folds to come close together. So if we look at the figure in the blue box and we focus on the part of the neural folds, we're actually able to see that the two neural folds are coming close together. And not only is the neural plate coming close together, we can also see the surrounding epidermis that is also coming close together. Now we can see the dorsolateral hinge point as this green structure, and there is also a change in the shape of the cells to allow the formation of the dorsolateral hinge point, but it is this dorsolateral hinge point that actually allows the neural folds to come close together. Now the final step is the closure of the neural tube. So the paired neural folds are brought close to one another, And this results in the formation of a tube-like structure that is under a layer of epidermis. So when we look at the small blue box, we can see that the epidermis is now very close together and a tube-like structure has formed right below it. And in the next small blue box, the neural tube formation is complete, and right above the neural tube is a layer of the epidermis. Now what we can see is there was cells between the epidermis and the neural tube. Those cells undergo or migrate away through delamination, and they actually will form the neural crest cells, which will form the parasympathetic nervous system. Just as a reminder, the neural tube will ultimately form the central nervous system. Now let us look a little bit at the formation of the hinge points. Now the formation of hinge points requires a change in the cell shape. So when we look at the cells that are in the neural plate, they have an apical part and a basal part. The apical part of the cells have to undergo constriction. Now let us look at the figure that's in the blue box, which is we're seeing is a sheet of cells. Now imagine that this was the neural plate cells. And to form a hinge point, they have to undergo a change in the cell shape to form a V-like structure. So the cells have to change from a parallel stage where you can form a certain portion of it that is narrower than the other part of it. Now note where the apical and the basal part of the cells are, and where we have the medial hinge point, the apical part has to narrow or constrict, while there is not much change in the basal part of the cells. This apical constriction is caused by localized contraction of actinomyosin. Now, during the hinge point formation, there is also basal thickening of cells, and if we look at the location of the nuclei in the medial hinge point, we can actually see that the nuclei are towards the basal portion of the cell, simply because it is wider. Finally, there seems to be more cell division occurring in the basal region, and this causes more cells to be present that automatically causes a bending and the formation of the hinge points. Sonic hedgehog is known to play an important role, and it is secreted by the notochord. So the sonic hedgehog made from the notochord which actually induced the formation of the floor plate in the neural plate. The foreplate is the one that will actually form the medial hinge point, and hence sonic hedgehog plays a role in inducing the cells to become the floor plate. Now what has been found out is that high BMP signaling prevents proper medial hinge point formation. So even if the floor plate cells have been induced, they are unable to form the medial hinge point when there is high BMP signaling. And this can be seen in this experiment that is shown in the figure, where in the first blue box, the cells are expressing a constitutively active mutant of the BMP receptor. This basically means that these cells will constantly be activating BMP signaling in them. Now these are the cells that normally would form the medial hinge point, and as we can see in this figure, we don't see the V-shaped structure, we simply see a straight line of cells, and hence they're unable to form the medial hinge point. When we look at the middle box and focus on those cells that are in the blue box, in this case, this is where we have normal BMP signaling present, so there is no manipulation occurring, and we can see the formation of the V-shaped medial hinge point. Finally, on the other hand, if we actually manipulate the cells so that they express a dominant negative version of BMP receptor 1, what happens in this case is BMP signaling is completely blocked. So when we look at a complete blockage of BMP signaling and its effects on the cells, we can see a medium hinge point being formed, but the medial hinge point is in a very exaggerated manner. And that is what is basically shown in the final figure in the blue box in the lower portion, where we can see that if you have very high BMP signaling, then there is no V-shaped form. When you have very low BMP signaling, you have an exaggerated point form, which does not allow the proper terminal steps of the neural tube formation. However, when you have intermediate BMP signaling, that is when we see the proper formation of the medial hinge point. Thus, for the cells to become the medial hinge point, sonic hedgehog is important for that induction of the floor plate. However, the actual formation of the medial hinge point requires intermediate BMP signaling. Let us now focus on the dorsolateral hinge point. Now in this case, noggin, which is a BMP inhibitor, plays an important role in the dorsolateral hinge point formation. Noggin is expressed in the neural folds and in the notochord transiently. Now when we look at this figure, we can actually see a wild-type embryo developing a proper neural tube where it has closed appropriately because it is able to form the correct dorsolateral hinge points. However, when we look at an embryo where noggin is not present, then because of the absence of noggin, there is a lot of BMP signaling, and this BMP signaling does not allow the dorsolateral hinge points to form very well, because of which the neural tube fails to close. And this is what we can see in the smaller blue box that is there, that is focusing on the neural tube and how it has failed to close. Now it is thought that by lowering or preventing BMP signaling, it actually causes the cell-to-cell junctions to relax a bit so that we can have the bending structures that are needed to form the dorsolateral hinge point. Thus, when we look at the actual different signaling molecules that are present during primary neurulation, we have the notochord making sonic hedgehog, and the sonic hedgehog induces the cells that are right above it to become the floor plate. And when we have intermediate BMP signaling levels, this floor plate can form a medial hinge point. On the other hand, when we look at the upper part of it, then we can see that noggin is present where we have the dorsolateral hinge points, and that way the BMP signaling is attenuated so that we can have correct formation of the dorsolateral hinge points. And thus, between sonic hedgehog's activity and BMP signaling, we are able to see how proper neurulation occurs. Now let's just pay a few moments on neural tube closure. Now in the case of amniotes, the neural tube formation does not occur through the anterior-posterior axis simultaneously. So the anterior part, which is also called as the cephalic or rostral part, tends to develop faster than the posterior, that is also called as the caudal part. So when we look at the neural tube formation, in this case, when we're looking at the anterior portions, we can see that the neural tube folds have begun to converge, but as we move towards the posterior end, we can actually see that the tube is just about to get formed, and thus there is a lag between the anterior and the posterior parts in terms of neural tube formation. Now the open end of the neural tube and the anterior portion that is shown in the smaller blue box is called as the anterior neuropore, and the one that is on the opposite side will be the posterior neuropore. Now neural tube closure in the case of mammals is initiated in different places. So in humans, the neural tube closure is initiated at 5 places along the anterior-posterior axis, and they all are shown in the figure as closure 1, closure 2, so on and so forth. There are many genes that are involved in neural tube closure, and right now we know that there are more than 300 of them. Now, to look at the mechanism of how neural tube closure occurs, it's usually by a zipping up mechanism that occurs in both directions. So This specific URL will take you to a video where you can actually see this zipping up effect occurring. Now, defects in the neural tube closure can result in conditions, and one of the most well-known conditions is spina bifida. However, depending on where we've had problems with neural tube closures, you can have different types of diseases. For example, in the case of defects in closure 1, you can end up with the condition called as craniorachis. On the other hand, if there is defect in closure 2, we can have anencephaly. If there is a problem at closure 5, that is when we have spina bifida. And that way, any defects in neural tube closure are associated with diseased conditions. Now, when we look at the molecules involved in neural tube closure, the cadherins are known to play an important role. Now, the neural tube eventually forms a closed cylinder that separates from the surface of the ectoderm. So there has to be some sort of driving force that is separating the tube from the upper epidermis. And initially, when we look at the cadherin expression, the entire tissue, the epidermis as well as neural tissues, produce E-cadherin. However, as neurulation occurs, the neural tissue switches its expression and does not make E-cadherin anymore, but it starts making N-cadherin. So when we look at the figure, here we are seeing that the neural plate cells are making N-cadherin, whereas the cells that are the presumptive epidermis are actually making E-cadherin. Now, E-cadherin likes to associate with E-cadherin, while N-cadherin likes to associate with N-cadherin. Thus, when we have the process of neurulation, when the neural groove is formed, we can see that the neural tube cells are coming together where the N-cadherins can associate with each other, and this allows the formation of the neural tube. On the other hand, the presumptive epidermis that were on both sides now see each other, and their E-cadherins will associate to form the upper epidermal structure. With that, we come to the end of primary neurulation, and we're going to look at the process of secondary neurulation. Secondary neurulation takes place in the posterior region of the embryo, and it occurs from a very different process. Now, the first process is the formation of a mesenchyme, which is normally formed from the ingressing ectoderm, which undergoes EMT to form mesodermal or mesenchymal tissues. And that is what is shown in the blue box. Now, these cells condense to form a medullary cord, and that is what is shown in the next step, where you can see these dense cells coming together to form like a dense tissue-like structure, which is the medullary cord. The next process is called cavitation, where you find these small luminal structures being made in the medullary cord, and they all tend to join together eventually to form a central cavity. And that is what we're seeing here, where in the medullary cord we can see small lumens or multiple lumens being formed, and then these lumens will ultimately widen and join together to form the neural tube. Thus, primary neurulation and secondary neurulation occur very differently. So when we compare the two, we can see that in the case of primary neurulation, we are seeing bending and then convergence to form a neural tube. Whereas in the case of secondary neurulation, we can see rearrangement of cells that are happening underneath that epidermal layer, and this causes the formation of the neural tube. Now, just as a reminder, primary neurulation occurs in the anterior part of the embryo, whereas secondary neurulation occurs in the posterior end of the embryo. Now, in between, we have the transitional region, and that is where junctional neurulation occurs, but we won't go into depths of how junctional neurulation actually happens. It's sufficient to know that the process involves components of both primary and secondary neurulation. Junctional neurulation is a relatively newly discovered phenomenon, and as we've seen before, it occurs at the boundary of primary and secondary neurulation. Junctional neurulation shows the hallmarks of both primary and secondary neurulation, and the mechanistic details that involve junctional neurulation are still being studied. Thus, to summarize, we looked at the different mechanisms of neurulation, and we focused on primary neurulation and secondary neurulation. We did look at some aspects of junctional neurulation and looked at neural tube closure.